Everyone, thanks for showing up bright and early today. I'm Bernard. I'm at Stanford. I'm in the Petrov lab. Today I want to, well, here's a protein, and I think like many of you, I'm interested in the evolutionary forces that have shaped the diversity in this protein across species. And okay, I guess I wasn't supposed to start yet. But I'm curious in how things like ecological shifts or developmental changes shape the fitness effects of all the bases in this protein. But even more fundamental than that, I think we need to first, before we start studying any of those things, understand base by base what the fitness effects of mutations are. And so what I want to talk about today is sort of methods we've been developing to try to do this in a way that's a lot better than what we have done before. So I think the best way to estimate fitness effects of new mutations using natural genomic variation data is from population genomic data. And here I'm just going to depict this as kind of the classic way in protein coding sequences, the contrast between non-synonymous and synonymous variation. And so this contrast, when you adjust it for the total number of possible sites at which these new mutations can occur, gives you a sense of the kind of natural selection that's occurring. So for a simple model of molecular evolution, like neutral theory, the only new mutations that can occur are neutral new mutations and strongly deleterious new mutations. And because of that, the levels of diversity, or the ratio of diversity in a population, reflects only the neutral variation that's segregating within the population. And so you kind of get this relationship or this ratio of non-synonymous to synonymous diversity that's fairly flat or insensitive to the allele frequencies within a population. And then, of course, this kind of leads to a similar level of substitutions. And so what this is kind of showing is that in the populations, this is where the diversity is flowing through the populations, and this is where natural selection is happening. And that is kind of the thing that's the most informative when we're trying to infer what the fitness effects of new mutations are. And we can fit more complex models. And so, there are slightly more complex models where we can have weakly deleterious new mutations that segregate in the population but never become fixed. And in that case, this ratio between synonymous and non-synonymous polymorphisms reflects the kind of gradual removal of deleterious variants from the population over time. So, when you're kind of early on and the things that are younger and at lower frequencies, they kind of more reflect the distribution of fitness effects of new mutations. And then as things get older and closer to fixation, it's kind of the effectively neutral variation that's left. And so again, in the absence of any adaptation, you end up with a substitution rate that sort of reflects these underlying processes happening at the population genetic level. So this is kind of the way that the field has done a lot of selection inference, again, from natural variation data for many, many, many years, decades. And methods to do this, including things that we've developed for this work, but I won't go into it too much because just for the sake of time. Take genomic data. And here I'm just showing you a sample or an example from Drosophila melanogaster. This is the whole genome. And fit some kind of model to this relationship or this ratio of synonymous and non-synonymous variation. And from that, you can try to estimate what the DFE or the distribution of fitness effects of new mutations looks like. And you can also estimate these more general things like the proportion of neutral new mutations or, conversely, the constraint on new mutations. And usually in a lot of natural systems, the substitution rates, or this DN-DS ratio, usually exceeds the sort of neutral baseline expectation, and a lot of that is taken as evidence for adaptive substitutions driving a good fraction of non-synonymous variation between species, and this is the basis of the McDonald-Kreitman test and its derivatives. One thing I want to note is that here, these are millions and millions of SNPs. This is the whole genome. And you need the common variants to really get that information about constraint. And so sampling deeply within a single species, like a lot of new modern genomic data sets, doesn't really get you any more common variation. So we're kind of left to lumping over a lot of heterogeneous elements in the genome. And because of that, we, I think, lose a lot of specificity. Like we get these numbers, and there's been all these debates about how natural selection acts across different species. And then you take all the genes in the genome, you lump them together, and you say 5.7% of new mutations are neutral, and 57% of non-synonymous substitutions are adaptive. What does that even mean? Is it the same for all of these genes, or does it, is there a spectrum? And I find that deeply dissatisfying because, again, like I was saying in the beginning, this really obscures the function that we know is there, and all these functional differences, and just kind of gives us a single number. And the reason why this happens is because polymorphisms are very, very sparsely distributed throughout the genome. So that protein that I showed earlier, again, it's not important what the protein is specifically, but it's an enzyme in the purine biosynthesis pathway. I just pulled some annotations here from the UniProt database, and I'm plotting the SNPs from the DGRP2 Drosophila melanogaster data set kind of in the space of this UniProt protein. So out of 700 residues, you can really see that there's 10 SNPs. There's 4 non-synonymous and 6 synonymous SNPs. When you're trying to understand how all these little, functional details translate to natural selection, you just simply don't have the data to do it because you don't have variation at over 99% of your sites. And so the idea that we had was that maybe we could try to improve the resolution of population genetic methods by adding polymorphism across many closely related species. And I think Drosophila is a perfect system to do this. It's high diversity. Things are easy to sequence. There's a lot of ecological diversity and other sort of features of this group that are useful to us. And so maybe instead of lumping a bunch of different genes across a single genome, what we can do is take the same gene across a bunch of different species, combine that data to learn something more about natural selection in a more precise manner. And to do this, we've been, for the last few years, developing methods just to do really, really cheap sequencing, taking advantage of liquid handling robots and amazing technicians like Hannah, who's here in the audience. And we've gotten the cost of genome assembly down to $150 per Drosophila genome, and for high coverage resequencing, it's about $30 an individual, all-inclusive. So that really allows us to go out there, collect any Drosophila species, collect population genomic data, and just start spamming this across the entire tree of the entire family. So right now we have another thing that I'm very proud of is that we're trying to do this in an open science manner and just really making the data available as fast as we can to the scientific community. And so right now we have, I think, about 500 species genomes, although that number increases every week. And we have 150 population genomic data sets at a variety of sample sizes, but we've kind of been sampling pretty much everything we can get, so it's a little haphazard. And there are hundreds more genomes and hundreds more population genomic data sets in our freezers. And so I'm going to gloss over this really quickly, but we do all this genome assembly, variant calling, do some basic QC, and then we use both whole genome alignments and then more precise alignments of coding sequences to try to get everything into the same coordinate space so that we can compare both substitutions and polymorphisms kind of in the same, for what I'm going to show is the melanogaster, the UniProt proteins. Okay, so when we have that amount of data, this is kind of the same protein from before. We only had 10 SNPs with Drosophila melanogaster. 99% of the sites were not variable. And that's not because these sites are under selection, it's just because we simply don't have the data to resolve anything. And so as we increase the number of species, we can slowly see that we start to fill up every single possible site with variation, and by the time we get to kind of our initial data set, 150 species, pretty much every single site has at least one polymorphism, meaning that we're able to apply some of the methods I talked about before, but at a resolution that is much, much, much better than standard single species population genetics. And so what I want to really start by saying is, okay, now we have this amazing data set, can we start inferring what the distribution of fitness effects looks like from gene to gene instead of just a single genome? And, I think this is kind of an obvious result, but yeah, selection is extremely variable across all the different genes. And so here I'm showing a plot of 3,000 very conserved orthologs across the family. Each column is a single gene, and then these different colors represent the proportion of new mutations that kind of fall into the categories of neutral, weakly deleterious, and strongly deleterious that I showed before. And so it's kind of a spectrum of Kimura genes on one end, where there's no segregating deleterious variation, and OTA genes on the other end. But what was, I think, also surprising, even in a species like Drosophila, about 20%, at least at the population genetic level, 20% of the genes are consistent with the original neutral theory. And then we can do the other kind of cool thing is that it lines back up to these sort of genome-wide estimates. If you look at the medians and the average of averages, it's not quite the same, but it's nice that there's this internal consistency in the data set and the methods. We can do the same game with looking at adaptive substitutions, and again, there's a huge spectrum of how much each gene adapts. And there are even genes that have no evidence for adaptive substitution, even in species like Drosophila, where adaptation is thought to be rampant. And of that, if you also compare it with the population genetic estimates, about 2.5% of these 3,000 orthologs are entirely consistent with the original neutral theory. And again, the sort of median rate of adaptation lines up with previous estimates. And so I think it's very cool here that what we're showing is that, sorry, I got a little dry throat. What we're showing is that it doesn't really make sense in this kind of modern era of sequencing and cheap and easy genome assemblies to argue about what the mode of natural selection is, but really what we need to do is go through the entire genome and estimate the heterogeneity in these processes properly. The last thing I want to leave you with is just, I showed at single gene level, but clearly even individual genes are very heterogeneous. The proteins have differences in function that differ from residue to residue. And kind of going back to this, because we have a lot of genomes, we can look at the variation in evolutionary rates, just DNDS, across all the different sites. And so this is a fairly constrained gene, but you can see that there's still a bit of variation across the protein, and we can zoom in a little more to where the region where the catalytic residues sit. And one thing that's obvious here is that right around the catalytic residues and a couple of other windows, there's extreme constraint. Every single species is the exact same across the hundreds of species that we've looked at. But you might think the rest of the protein looks fairly unremarkable. But what if we layer the population genetic data on top of that? So we then can see these windows where there's no variation at the population genetic level across all of the hundreds of species that we've looked at, but where there's substitutions, non-synonymous substitutions. And so these are some clear instances where we're pretty confident that this is adaptive evolution. And it's because we have this contrast of substitutions and polymorphisms that we're not affected, or that this process of adaptive substitution does not obscure our inferences. And so the very, very last thing is that these patterns also don't just make sense in linear space, but they cluster into 3D space. And so if you look at where these constrained windows occur, they occur directly around the catalytic residues in the protein structure, and the adaptation occurs at an intermediate distance. And we think that maybe this shows that adaptation has to occur close enough to affect the function of the protein, but it can't occur directly at the most important sites. And so Alan has a poster, and I'll just skip to the acknowledgements. And there's just a ton of people working on this, and this would not be possible without them. I'm starting my own lab. Send me an email if you're interested.
First, I would like to thank the organizer for putting this symposium together and giving me the opportunity to present this recent data that we obtained about the distribution of fitness effect of synonymous, non-synonymous, and regulatory mutations. And so our first motivation to put this experiment together was to get insight into the fitness effect of synonymous mutation. So initially, synonymous mutations were considered as all and always neutral. They were even called silent mutation. But there are recently evidence that some synonymous mutations have a phenotypic or fitness effect. And there has been quite a lot of work on the mechanisms that can explain these phenotypic effects. So the main mechanism, or the one that has been most described initially, was the effect on the translational speed and accuracy, depending on the codon usage and the fit of the codon usage with the codon usage of the species. There are now quite a lot of work about the effect of the nucleotide sequence on the messenger RNA structure, secondary structure around the start codon, and that's conditioning the access of the ribosome to the ribosome binding site. And finally, more recently, there's been quite a lot of work on the effect of a synonymous mutation on changing or introducing DNA motifs that carry a signal, for example, splicing signals, promoters, restriction sites. So there are experimental evidence of this fitness effect of synonymous mutations. So there are, for example, many cases of individual synonymous mutations which have a strong effect in experimental evolution approaches. And then there are also evidence that are coming from synonymous variant approaches. So synonymous variants are a collection of DNA sequences that encode exactly for the same amino acid sequence, but they use different codons to do that. And so you have an example of a collection of synonymous variants of a gentamicin resistance gene on the left of the slide, and you can see that there are many differences between them, although they encode for the same amino acid sequence. And then we can measure some fitness or phenotypic traits. So on the right, you have the resistance conferred to gentamicin of these different variants across 3 species that are color-coded. And you can see that depending on the synonymous variant, the resistance conferred is very different. But from these 2 kinds of evidence, we don't know anything about the distribution of the fitness effect, because in the first case, we have the effect of individual mutations, and in the second case, we have variants that differ by many positions, and so we don't know if the effect we see is the combination of small effect at each position, or if the majority of the synonymous mutations are neutral and some of them have a large effect. So that's the first question we wanted to ask. And then in experiments we have conducted in my group, we have experimentally evolved synonymous variants in selective environments, so mainly in antibiotic. And the main results that we had is that we had generally no mutation in the coding sequence, either synonymous or non-synonymous, but mutations that were affecting the expression level of these genes. So we had mutations in the cis-regulatory region of the gene, and more recently we also had very huge variation in the plasmid copy number, so because the gene was carried by a plasmid. And so this kind of triggered the idea that it's easier to evolve by regulatory mutation, to adapt by regulatory mutation than by synonymous mutations. And so we were interested in comparing the distribution of fitness effect of synonymous mutations and regulatory mutations. So that's the first 2 questions, and then we added the last one, which was asking the environment dependence of these fitness effects. So how did we try to answer this question? We use an approach which is quite close to deep mutational scanning. So deep mutational scanning is creating a collection of DNA sequence in which at each amino acid position is replaced by any other possible amino acid. So we applied the same logic, but at the nucleotide position. So we created one single point saturation mutation library, which means that for each nucleotide position, we had variants with the 3 other possible nucleotide positions. It's actually mimicking the real mutational process. So we had all the mutants that were on one mutational step of our wild type sequence. And by doing that on a gene and its promoter, we created a collection of regulatory mutations, synonymous mutations, and non-synonymous mutations. And by computing them in mass and measuring the fitness by deep sequencing at the beginning and at the end of the competition, we could derive the distribution of fitness effect. So we did that on a gene called SHBLE, which is a bleomycin resistance gene. So it's a resistance gene, and it's also very short, so it's easier to make all the variants. The bleomycin is a glycopeptide antibiotic that is acting by inducing DNA breaking. And it's coding for a protein that dimerizes, as you can see on the picture, and the resistance is provided by sequestration of molecules of bleomycin within the two grooves that are formed when the protein is dimerized. So now we have the full experimental plan. So we have a library of more than 1,000 single nucleotide mutants, and that was cloned on a low copy plasmid. We have done competition with the wild type in excess. Each competition was done in triplicate, and we measured the fitness at the beginning and at the end by the ratio of the abundance at the beginning and at the end. And we did that across 5 concentrations of bleomycin, of antibiotics. So first in absence and then 4 concentration. The highest concentration is very close to the MIC. And so that's the whole set of results. So you have on the different lines the different concentrations, so the environmental conditions, and in the column the 3 categories of mutation. So what we can see on the global picture is that the synonymous mutation on the right in green have a distribution that seems quite similar across the different concentration, whereas the non-synonymous and the regulatory mutation have a distribution of fitness effect that is extending to the left, so to negative fitness values when the concentration is increasing. There are some small exceptions here in the synonymous mutations, so we have a few number of synonymous mutation with a strong effect. If we compare this distribution by pairs within each concentration, we see that they are all significantly different, except the non-synonymous and the synonymous mutation in the absence of antibiotic. If we now fit this distribution, the important information on this slide is that for synonymous mutation, we have always a unimodal distribution, whereas for the non-synonymous mutation, it's mainly unimodal in the absence of antibiotic. But then when the concentration of antibiotic is increasing, it's getting bimodal and then trimodal. So this is another way of showing this extension of the distribution to the left. And for the regulatory mutation, there is a single peak for all concentration except the highest one that is quite centered around 0. But then at the highest concentration, it's dividing into 2 peaks with a positive one and a negative one. If we now try to see a bit more what's happening with the environment dependence, so here it's the same data, but represented as the fitness effects against the antibiotic concentration for the 3 categories of mutation. And so we can, so on each of the 3 graphs, you have all the mutations represented, but the one, so each category is highlighted in one of the graphs. And it's visually quite clear that the non-synonymous mutation have a strong environment dependence, whereas the synonymous one are in majority stay around neutrality when the concentration is increasing. So if we now fit simply a linear regression for each of the mutation for the effect on the across concentrations, and we look at the proportion of this fit that are significantly positive and significantly negative, so that's the value you have at the bottom of the slide, you can see that for the positive one, the proportion is quite similar across the 3 categories of mutation, whereas for the negative one, we have very few for the synonymous mutations, a bit more for the non-synonymous mutation, and close to 45% of the regulatory mutations that have a negative relationship between the concentration and their fitness effect. So now we have tried to understand a bit what was the origin or the mechanisms for the effect of the synonymous mutation. And we tried to see whether we could detect a signal of an effect on translation speed and accuracy. So for this, we used these coefficients called WEG, which is the coefficient that is used when we are calculating the CAI, the codon adaptation index. It's basically looking at the frequency of the focal codon compared to the frequency of the codon of the same synonymous family that is most used in the species. And so we calculated delta, which is the difference between this coefficient for the wild type and for the mutant we are looking at. So for each of the mutants, we have a delta WIG, and we try to correlate that with the fitness effect. And there is absolutely no correlation between this index of codon match and the fitness effect. So we have, for now, and in this data set, no cues of fitness effect that is linked to the codon usage match. So something else we did to try to understand where the fitness effect of the synonymous mutations were coming from was to plot the effect of the mutations along the nucleotide sequence for each of the concentration. So here you have both the synonymous and the regulatory mutations. And what we can see on this picture is that when the concentration is increasing, we get strong synonymous mutation effects close to the start codon. And actually, these strong effects are mirrored on the regulatory side, also close to the start codon. So that could be that it's linked to the secondary structure of the messenger RNA around the start codon. So by changing one position, you change the conformation and you change the accessibility to the ribosome, so the level of transcription, and so the level of expression of the protein, which would explain why these mutations are sensitive to the environment, because they became a lot more negative when the concentration of the antibiotic is high. So to wrap up for now, so we have seen that the distribution of fitness effect of synonymous, non-synonymous, and regulatory mutations are different at all concentration, except the case of synonymous and non-synonymous in absence of antibiotic. Of course, this is true for the system I just presented. I don't want to generalize to everything. Regarding the environment dependence of fitness effect, we have seen that this environment dependence is lower for synonymous than for non-synonymous mutation, and lower for non-synonymous than for regulatory mutations. And the fitness effect of synonymous mutation seems to be more linked to their effect on mRNA secondary structure than on their effect on translation. And that's actually a result that has been also found by other methods in other studies. There are many more things that we can look at in this data set, but I don't have time to present them. So, for example, we can look at the correlation of effects between environment, and I can tell you that they are quite strong for the non-synonymous mutations and not that much for the other categories. We can also focus the analysis of the distribution of fitness effect on the positive effect mutation, because there are the mutations that are likely to be involved in adaptation to the environment, and so look at which categories are overrepresented, in particular in the outliers, and so the mutants that are more likely to raise in frequency. We can try to look at the origin of the fitness effect of non-synonymous mutations, in particular looking at what we know about the rate of substitution between amino acids and the difference in the chemical properties of the amino acid. And we actually have also we have nonsense mutations in these data sets. We have really done all the mutations, so including mutations from the start codon, from the stop codon, and to the stop codons. And so we also have, I think, interesting data on this. So finally, I would like to thank Martijn Callens, who actually did all the lab work and the data collection. Colleagues who have helped either in the lab or by discussion of when setting up the experiment, but also when interpreting the results, and funding, and you for your attention.
Thank you everyone for coming to my talk. And the work that I'm about to present was done with collaboration with Olivia Gosch, who is also here. So the big question that we are asking is, what determines the long-term success of a genotype that competes with many other genotypes in a population? And thinking about this question, the first idea that probably comes into mind is, well, probably initial fitness should matter a lot. If you start with high initial fitness and high initial frequency, you can just outcompete everyone else, and take over the population. But even if the competition happens on more long-term evolutionary timescales, adaptive mutations will be more likely to arise on high fitness backgrounds and less likely to disappear due to drift. So high fitness genotypes should still outcompete and become so-called winners. So does it mean that if you start with low fitness, you're totally doomed for extinction and there's no hope for you? Maybe not. We see from observations in natural populations and colonial populations in a lab that sometimes organisms can leapfrog. So when an organism starts with a low fitness, it can still somehow outcompete high fitness counterparts. And this leapfrogging could, of course, happen to some extrinsic outside factors, such as change in environment. But it could also potentially happen due to some internal ability to leapfrog. For example, due to its ability to interact with other organisms and like poison everyone and take over. Or maybe it has high mutation rate and thus has more potential for evolvability. And so the two specific questions that we want to ask with our experiment is, first, to what extent specifically, exactly, does initial fitness predict long-term evolutionary success? And if it's not 100% prediction, then within a species, do different individuals differ in their internal ability to leapfrog? And so we started to think, okay, well, what kind of experiment do we need to do to answer these two questions? And we figured that, first of all, we need a system that will have a large collection of diverse individuals, and these individuals should be diverse both in their genotypes, but also in their starting fitness, such that each genotype has an ability to show off its leapfrogging potential, if, of course, it's real. Second, we need to be able to measure their short-term fitness really well so we can make qualitative predictions about their longer-term fate. And lastly, we need to evolve them for a long period of time and in many replicates such that we can observe winners taking over the population, and also we should account for inherent stochasticity of evolution. Thus, we need many replicates. And you probably can guess where I'm going with this. We have a system like that in our lab that checks all the boxes. So the system is a collection of wild Saccharomyces cerevisiae strains that we got from our collaborators from France. And it includes yeast strains from all over the world. And not only they differ in their geographic location, their geographic origin, they also differ in their ecological origin. So we have yeasts from wineries, bakeries, breweries, clinical isolates, wild yeast. If you know anything that yeast can do, we probably have that in this collection. And another amazing feature of this collection is that all the yeasts are barcoded, so they have a random piece of DNA integrated in the same location in their genome, so we can amplicon sequence this barcode and track their frequencies over time. And so what we decided to do, evolution with the pool of these natural isolates and transfer them every day into fresh media in a 10 microliter volume culture and 1 to 250 dilution. So it's roughly 8 generations per transfer. And we did that in 5 different environments that are all based on synthetic complete media, but they differ in their chemical composition. And we also did 20 replicates per environment because, as I mentioned before, we figured that this experiment should be done in enough replicates. So timeline-wise, we started this experiment in the January of this year. And we did transfers every other day since a few days ago when we flew to this conference. And so then we sequenced the first 5 time points, the first 5 transfers, to infer short-term fitness of our natural isolates. And so the data that we got from this was looking something like this. So on the x-axis, we have time points in transfers, and on the y-axis, we have frequency of each lineage over time. And they're colored by the estimated fitness, which the way we define it is proportional to the log of frequency change of each lineage over time. So it's roughly the slope of these lines. And so then we can look at distribution of fitness effects of all these natural strains across different environments. So in this plot, each dot is an individual strain, and they're colored by their fitness at M3. And I hope you can appreciate that the initial fitnesses are shuffled enough across environments, and this is what we wanted to achieve. So, as I said, we can control for their initial fitness when we look at their longer-term evolutionary fate. And so then we reasoned, well, okay, if the initial fitness is the main predictor, is the main thing that matters for longer-term success, then among the winners of our experiment, we should only find the top fit, the most fit strains. And you know, this is still experiment in progress, but we have already sequenced some of the later time points, 9, 23, and 63, and I want to share some of this data with you. So here is extended data for one of our replicates in one of our media. Now the x-axis is not linear because it includes this later time points. And here you can see that there's 3 lineages that reached high frequency at the last time point that we sequenced. And we can look at these lineages more closely. It seems like they were doing pretty well in the beginning. They're doing pretty well in the end. So it seems pretty expected result based on our initial fitness values. But if we want to quantify their long-term, their long-term outcome, we should really look at this top right quadrant. So just the last time points and just the top, the most frequent strains. And we used an artificial threshold of 5% frequency or more. And so in this quadrant, we have 5 different strains. And we can look back at their initial fitness and see how they were ranked relative to other strains. And so the BEP strain was ranked number 1, and it is doing the best. AMA strain was number 2, and it is second best, and so on. And so I don't have time to show you data for all our replicates, all of our environments, but believe me when I say that in 4 out of 5 environments that we had, in all 20 replicates, the strains that reach 5% frequency or more are always among the 5% top fit strains in that specific environment. So it seems like most of the time, initial fitness is a pretty good predictor of what happens longer term. But we have one environment in which this is not exactly the case. That was M3 minimal media with 2% ethanol. And in this media, we have some of our replicates that look something like this, and I'm going to highlight the strain that basically fixes in these 2 replicates. It's the strain called BIE. And if you look at the initial slope of the strain, it wasn't that great, but somehow it reaches to a very, very high frequency at the end. And in order to get the sense of, you know, whether that is actually as surprising as it looks by eye, we ran a simulation in which we simulated the longer-term time point frequency at longer-term time points for each strain, just taking into account their initial fitness, assuming no evolution, no interaction, nothing interesting that could potentially happen. And so what we saw in our simulation is that in 1,000 times of the simulation, this BIE strain dies out, goes extinct every single time. But in reality, it does not go extinct in any of the replicates, and instead, it, you know, sometimes it becomes really, really high. So what is the BIE strain? It's a wine strain from Italy. It is used in production of white Italian wine. And we don't really know much more about it, and it doesn't seem to be special in any other way. But we figured that we can look more in our data to see, you know, whether this strain actually has an internal leapfrogging potential or not. And so we reasoned that if it does have an inherent ability to leapfrog, we should see doing it in many environments, not in just one. And when we look at that, it, not as dramatically as an M3 ethanol, but in other environments, it consistently performs better than we would expect purely based on its initial fitness. And so with that, I'd like to conclude what we have so far. So to what extent does initial fitness predict long-term evolutionary success? Well, yeah, to a very large extent, and most of the time, strains that end up with more than 5% at the end were among the top fittest strains in the beginning, and this is consistent with some with other data by other people, consistent with common sense, with cancer data, and so on. And then our second question was, within a species, do different individuals differ in their leapfrogging potential? Well, of course, it is too early to say yet, but we have one interesting candidate strain that we would like to investigate more to see whether this is this, or maybe this is something else. So our next steps, we want to sequence more time points, including the last time point, T91, but also more high resolution in between. We want to whole genome sequence winner clones in each of our environments to get a sense of the genetic targets of their adaptive mutations. And also we want to integrate high diversity barcode in a few candidate strains, including the BIE strain, and test whether they have an inherent ability to leapfrog in a more detail. And with that, I would like to thank my lab, the Petrov lab, Olivia, Dmitry. I would like to thank our collaborators who provided us with the natural yeast collection, my funding sources, and you for your attention.
There's time for a couple of questions. In the back, there was the first that raised your hand. Yes?
So I was, I was curious, thank you for your talk. I was curious, did you look with, especially the most successful yeast strains, did you try to quantify by growing them in isolation what might be driving their initial higher fitness?
We have not done that yet. We have another person in our lab, Shaylee, who is doing growth curve experiments on them. So we're definitely going to look into, we're going to ask for her to collaborate with us and compare her growth curve data with our data. But at the first glance, it seems like growth curves look more or less the same for most of the strains. But yeah, that's a good idea.
Really fantastic experiment. Do you have some ideas about what pattern you could observe in the driver mutations that would give you a sense of mechanism for the leapfrogging, for vulnerability?
Yeah, so this is what we want to look at when we whole genome sequence the strains, and I think really interesting thing would be to look at whether the mutations that this BIE strain acquires make it in some way phenotypically or genotypically more similar to the strains that had high fitness in this environment to start with, and whether this is an example of some kind of convergence, or maybe it could be something totally else. Maybe it poisons everyone. We don't know yet, but we are very excited to do that.
How much is known about the fitness landscapes in these environments and how the difference in the stress of these environments? If you have 4 or 5 environments are relatively less stressful, right? Maybe the fitness landscape is really simple and really high proportion genotypes can easily increase the frequency, but something more stressful if many of these other genotypes are close to a local peak, but there's a great peak to be had, right? Now you have an opportunity for the less frequent ones, right, to kind of explore that landscape and actually find the higher peak.
Yeah, so for the M3 environment, we have pretty good understanding what could be happening because of we had a high diversity barcoding experiment, Levy-Blundell, that was in that specific media. But it's interesting that you said this question because we also had other environments that I didn't include in my talk. For example, SC, which seems much richer media in which yeast should be happier. But in that strain, just one, yeast takes over very quickly within the first few transfers, and it seems to be doing much better. So we couldn't find any correlation between like our understanding of how stressful they are and how many screens are left.
All right, hello, my name is Jose Aguilar-Rodriguez, and today I'm going to be talking about one of the most exciting projects I've been working on during my postdoc. So let's get started. One of the long-standing debates in population genetics pertains to the question of the nature of standing genetic variation, about the fitness consequences of segregating variation in natural populations. And there are two opposing models about this that I'm going to explain using this allele frequency spectrum diagram. So here I'm representing the proportion of variances as a function of their frequency in populations. So we have the neutral theory. It's actually an extension of the classical theory by Hermann Müller that proposes that variants that are at low frequency tend to be in a mutation selection balance where purifying selection is keeping them low, and they tend to be deleterious and have a functional consequence that we can measure experimentally. And neutral theory proposes also that like variants that are more common in the population and have like larger frequencies tend to be neutral or nearly neutral and be non-functional. This is kind of the null model, and the alternative hypothesis proposed by Dobzhansky in the 1950s is balance theory, where a substantial fraction of like these common variants actually have a fitness effect, are maintained at high frequency without reaching fixation due to some balancing selection mechanisms, such as, for example, local adaptation. The debate is still ongoing in part because of limitations with the data that we currently have. So there are numerous population genetic studies using population genomic data that have shown that the distribution of non-synonymous variants, those that change the amino acid of a protein, are indeed shifted towards lower allele frequencies, suggesting the action of purifying selection. But we still find non-synonymous variants at large frequencies in populations. The neutralists propose that these are variants that are tolerated by the proteins, but the jury still is out without experimental validation. We also have then experimental approaches coming from quantitative genetics, such as QTL mapping, that lack the spatial resolution in most cases that we need, so they don't have single nucleotide resolution. And we know that most of the variation in natural populations are actually like single nucleotide variants. But in the last 5 years, recent advances in precision genome editing have made this question tractable at a scale that was unprecedented before. So for a very long time, we had precision editing, which is just modification of the genome at single nucleotide basis. But they had very low editing efficiencies, between 5 and 20%. So it was not enough to do high throughput genome editing. But in the last 5 years, there is several methods, including CRISPR, which is the one I'm using, that have very high editing efficiencies, between 80 and 100%. So now we can do massive parallel genome editing at single nucleotide resolution, where we can introduce tens of thousands of variants. And in my postdoc, in collaboration with the Fraser Lab at Stanford, I have worked on CRISPR barcode, which is a method that allows to introduce 2 edits at the same time in a cell. So we can introduce a mutation of interest that we want to measure its fitness. And also in the genome, we can introduce a genomic barcode. And because we know this association between barcodes and edits or mutations, we can combine them in a flask, and we can compete them, make them compete, and we can track them by sequencing the genomic barcodes, the frequency of these mutations in the population. And from this, we can estimate selection coefficients. And we usually have hundreds of barcodes mapping into the same mutation or variant, which allows to measure fitness in a very precise way. Okay, so this is the method, and this is the system I decided to study. So these are 2 core eukaryotic signaling pathways, the RAS-PK pathway and the TOR pathway. They are highly conserved from yeast to humans. And the reason why I chose them is because evidence from experimental evolution, as well as cancer biology, suggests that these pathways are highly important for adaptation. So it's not unlikely that we are going to find functional variation in these pathways. So like, for example, in humans, more than 1/3 of human cancers are actually caused by mutations in RAS and TOR. And I'm working on yeast and Saccharomyces cerevisiae, so the type of genetic variation is coming from more than 1,000 strains that I'm representing here in this phylogenetic tree. The different colors represent the different ecological origins of these strains. So we have wine strains, beer strains, strains from the wild, clinical strains as well. And we find like 14,000 single nucleotide variants in these in the genes of these pathways, both in the coding and non-coding regions. And using CRISPR, barcode, I was able to successfully edit 9,500 out of these variants. So there is a limitation in the method, is that you need a PAM sequence near your variant of interest in order for Cas9 to cut. But it's still a large fraction of this natural variation that I was able to introduce. And I compute them in a minimal media where glucose is limiting. I did transfers with a 1 to 20 dilution, so 4.4 generations every transfer for a total of 26 generations, so not enough for de novo mutations to occur. And I had 3 replicates per experimental condition, and I had 4 experimental conditions. So the experimental conditions was like transferring the cells every day, every 2 days, every 3 days, and every 5 days. And the reason is that these growing in a flax have this growth curve in black where they go through different phases of growth, lag phase, fermentation, where they ferment glucose into ethanol. Then when they run out of glucose, they go into respiration, and finally into stationary or starvation phase. And by changing the length of the cycle, I'm changing the time that the cells experience these different phases of growth. And we know from experimental evolution that there are usually fitness trade-offs between these different phases. So for example, variants that are very good at fermentation tend to be very bad at stationary phase and vice versa. So we wanted to see if this also happens in natural variation. All right, so let's see some of the first results. So this is for the 2-day condition, and here I'm plotting the fold change in frequency of the top 10 most deleterious variants in blue. And you can see that they are going down in frequency. And reds, I'm indicating the frequency trajectories of lineages that are barcoded but not edited. So these are neutral, they have neutral fitness, and this is kind of our baseline. And then these are the top 10 most fit variants that are increasing in frequency, and actually one of them is oncogenic mutation in humans. And from this type of frequency trajectories, we can estimate selection coefficients, we can estimate fitness, and this is the distribution of fitness effects for this condition, the 2-day condition. And the first result is that most of the variants are actually neutral. Only 15% of the variants are significantly non-neutral in at least one experimental condition. And because we were able to measure the editing efficiency as very high in this experiment, as 85%, while there might be false negatives among these neutrals, we really believe that most of them are going to be like biologically neutral or nearly neutral in these conditions that we measure. All right, so I'm going to focus on the set of like 1,400 non-neutral variants. And the first observation is that most of them are actually non-synonymous variants. So more than 75% of them are changing the amino acid of the proteins. And what are they doing in these proteins? So here I'm comparing the non-neutrals in blue with the neutrals in orange. And they are actually changing more conserved residues. So this is the CIF score. The lower it is, the more conserved the residue is. They're also happening here in plotting packing density, which is just the number of amino acids around in 3D space around a particular residue. So non-neutral variants tend to happen in more packed regions of the protein, also in less accessible to the solvent. So this is solvent accessibility. This is delta delta G. They tend to be also more destabilizing. So it looks like these non-neutral variants tend to be also like functional and changing important protein properties. What about their allele frequency? They also tend to be at lower allele frequency in the population. And if we look at the age of these variants, they tend to be younger also. So just to recap so far, the data, the results that we have found seem to support the neutral theory in the sense that most of the variation is neutral, and the ones that are non-neutral tend to be at lower allele frequencies, they tend to be functional, and they tend to be deleterious. But remember, like balance theory proposes that there are meaningful fraction of like common variants that should have a fitness effect. So what's going on? What happens to these? So I'm plotting the maximum absolute experimental fitness across the 4 conditions as a function of the number of strains that carry a particular variant. So this is just like another way of representing their frequency in the population. And you see that there is a large number of like highly frequent variants which have high fitness. And we think that these are under some kind of like balancing selection. And today, for the sake of time, I'm just going to walk through one of these examples. So this is a mutation in SCH9. This is the vector kinase of the TOR pathway. This variant is happening in a highly conserved part of the protein, and it has this dramatic increase in fitness in the 2-day condition. And if we look at the phylogenetic distribution of these variants, they cluster within the wine clade in purple. And if we zoom in, they appear in this subclade. And these are actually clinical strains. So these are infecting humans. And we think that this variant could be playing a role. So like, I'm doing like a reconstruction of this variant to measure its fitness, for example, at high temperature, because there is some evidence that null mutations in this enzyme, in this kinase, are actually increasing thermotolerance of yeast. And then if we look at the fitness of this variant across the environments, so it has this positive fitness effect in the 2-day condition, in the 3-day condition, in the 5-day condition, but in the 1-day condition where fermentation is dominating, actually has a negative fitness effect. So this like fitness trade-off also suggests to us that like this could be a case of like local adaptation. So yeah, the reality is complex. The nature of standing genetic variation is complex, and we find evidence for both theories. So of course, these are two opposing simplifications, two opposing extremes, but I want to convey to you that using this type of methods, we can like start quantifying the relative contributions of these two models to, you know, like standing genetic variation in the wild. So just to summarize, show you how we can use these high throughput precision genome editing methods to study tens of thousands of genetic variants in full biological pathways. And in particular, in the pathways I study, find that the small fraction of non-neutral variants tend to be non-synonymous, not to affect different important protein properties, and be deleterious and at low frequency, but there is still a substantial fraction of variants at high frequency that show fitness trade-offs. I'd like to thank my labs, especially Olivia, for the fitness measurements of my frequency trajectories, and Cian for help with the experimental method. And yeah, thanks for doing it. Yeah.
Really nice work.
Thank you.
You engineered in single SNPs.
Yes.
So far you're blind to epistatic effects.
That's right, yeah. Yeah. One possibility could be, I mean, to extend it, could be to repeat the experiment in different backgrounds. And the problem is that like I'm only able to edit 2 sites, and I'm using one of them for a barcode. So like also, you know, another possibility would be 2, but I don't know. Yeah.
Thank you again for hosting this great symposium and for inviting me. My name's Carl. I am a grad student at Vanderbilt, and I'm excited to share some of my work with you today on Bayesian analysis of longitudinal fitness screens based off transposon mutagenesis. So I work on E. coli, specifically bacterial adaptation to starvation. So I want to begin by refamiliarizing you all with the kind of general bacterial growth curve. So first, there's this period of exponential phase, the stationary phase. You have rapid growth, you have rapid swings in pH, you have a rapid decrease in preferred carbon, other food sources. This is in LB, so an amino acid-based media, but a rich media. Then the cells hang out in stationary phase for a while, and then they have rapid death in what is termed death phase. And then you can see an accumulation of this dead cell material, or necromass. And then you enter long-term stationary phase, where the cell density is constant for a very long time, but this is actually masking these dynamic subpopulations that emerge and grow and die at roughly equal rates. And so a characteristic of this 3-phase growth regime is that each phase favors opposite growth strategies. You have initially growth, and then it favors survival, and then it favors growth, but also in a very stressful environment. And so it's kind of both, but also different. And a lot of this is reinforced by E. coli regulation and physiology that has many levels of antagonistic pleiotropy that will direct kind of E. coli life towards one of these phases of either growth or survival. And so my primary focus here is to identify genes that are adaptive in this complex environment. And an excellent tool for this is a high-throughput genetic screen using transposon mutagenesis. So in particular, I'm using this bar-coded library created by the Deutschbauer and Arkin groups a while ago in E. coli. And the method is called randomly bar-coded TN-seq, or RBTN-seq. So here I take this library where each cell, it's hard to see here, each cell has one gene knocked out by a transposon, and then you pool it together. So collectively, your pooled library has every non-essential gene knocked out multiple times. And so typically this is done as an endpoint assay, and it's simple to calculate fitness from beginning and end and make a nice volcano plot like this, and then get some kind of test statistic like a TE score here. However, kind of classical statistical tests like this are much more complicated in a longitudinal study with hundreds or thousands of barcodes. And while you could do a million t-tests and compare over time, sorting through the significance between genes across time would be very difficult to interpret. And so I had to turn to specifically a modeling-based approach where I could infer fitness for each barcode over time. And so the key to this was kind of Bayesian inference, which briefly, just a high-level overview, allows you to infer fitness with confidence intervals, not just p-values, and it can account for noise, and sequencing experiments are extremely noisy. But it's a very large learning curve for kind of your everyday stats practitioners, and it's also pretty computationally intensive. And so I was looking for a method that would allow me to use Bayesian inference on all of my barcodes over time, but it didn't exist. And so I had to create a pipeline, which I call here Bayesian RBT-seq Analysis Kit, or bracket, which allows you to kind of plug in the data from this specific and very widely used TN-seq library, and then use Bayesian inference on it almost as simply as if you were just using the standard pipeline. So briefly how it works is you sequence and count your barcodes. You normalize fitness to neutral genes, which I have defined as one benefit to this library is that it's so widely used, there's a ton of published data on it. And so I could pull out one study, Price in 2018, grew this library in almost 100 unique conditions. And if you look at the barcodes with the lowest fitness over all of those conditions, they have virtually zero fitness effect, measured here by the root mean square of fitness. And so I can take these neutral genes N and then normalize each gene to those neutral genes, just taking a log ratio of your gene barcode to your neutral barcode. And so this log ratio transform also has a few very specific benefits to it mathematically. First, since sequencing data are inherently compositional, the variance varies depending on the actual count. And so the log ratio transform fixes this and ameliorates that heteroscedasticity. And second, the normalizing to a defined set of genes rather than just the distribution average has a couple benefits in that it both centers the distribution, but also makes it so your results are easily biologically interpretable as being in reference to these neutral genes rather than some kind of less well-defined center, mean, average, whatever. And so then I calculate selection rate over time for each gene. This is just your standard selection rate formula where you have your log ratio of a gene at time t over the previous time point, and then you subtract your reference genotype, which here is the average of the neutral genes. And then just for completeness, you can rearrange the terms to nicely fit in your log ratio transformed values as well. And so I can go back then to this huge data set and also use it to parameterize my priors. And so that makes it a lot easier since you have all this data, but from a different experiment, you're not really peeking or double dipping on your data. So finally, I combine all these into a model. Since I only have 3 time points here, I'm just doing piecewise linear regression. And here's one example gene. I'll tell you later what it does. But FIMB knocked out by a transposon is not as fit as the neutral genes at day 0 to 1. And then it is more fit from 1 to 4. And then less fit from 4 to 10. And so you get these 2 nice segments just modeled by your linear regression here. And one benefit is to the approach of inferring fitness is you can also calculate other statistics based off of your inferred values. And so since this selection rate actually measures selection over time, if you multiply by time, you get these areas that you can then add together as kind of a summary of your change in abundance over the entire experiment. So FIMB is less fit, less abundant after the whole 10 days here. And so here is all genes ranked by their log relative abundance stat. And you can see roughly 400 are either more or less fit in that these horizontal confidence intervals do not overlap with 0. And so these results are in general consistent with what we know biologically. One thing to note, these values are about an order of magnitude lower than what you get out of your typical frequentist TN-seq analysis. And I interpret this right now as large clonal interference when you're co-culturing 70,000 genotypes all at once. And I also expect that simulations will show that since the data are so noisy, taking just a simple summary like a mean will still greatly overestimate the fitness effect of each gene. So to kind of gut check these results, I looked at what's called choke point enzymes. These are enzymes that are uniquely use or produce a metabolite and are also highly connected to other metabolic pathways. So one example I show here is just purine de novo biosynthesis, and these three enzymes in red are these so-called choke point enzymes. When you look at the predicted fitness of many choke point enzymes, you can see that the predicted mean fitness is well below 0 for each of them, or for most of them, I should say. And I have the probability, it's kind of small to see, but that each is below 0, shown numerically below each distribution. You can combine all these probability densities then into this large density, and so interpreting this means that there's a 86% chance that the average of all of these genes is below 0, which is pretty high chance, pretty concordant with what we know from previous results. And so what is the opposite of a choke point enzyme? It would be a redundant enzyme. So here's the enterobacterial common antigen biosynthesis pathway. I have some other evidence showing that disrupting ECA synthesis is deleterious in this system due to a kind of overzealous misfolded protein response. And so, but you can see that these two steps in the pathway actually have redundant enzymes that can both catalyze this reaction. And so if you knock out any one of them, they can be compensated by their partner. And so you can see here that the unique genes in blue are deleterious when knocked out, while the redundant enzymes are closer to neutral, still a little deleterious. But again, you can combine these values into probability distributions, and this gives you kind of a clearer, more empirical view of this hypothesis. And you can see there's a 37% overlap in the distributions, showing 37% probability that they are the same. And the complement of that then, 63% chance that these two distributions are different. And so 63% chance, not super high. I don't know if I would bet my house on it, but it is at least high enough that I would probably follow this up in the lab and do some knockouts. And so this is a great way to kind of generate hypotheses and get confidence bounds on what they could be. Now, just one last kind of vignette on this. Here's the FIM operon with my favorite gene, FIMB, that I've shown you before. The FIM operon regulates Fimbriae in E. coli, which are one of the major adhesins that form biofilms. And so there are two regulation genes, FIMB and E, and then kind of the structural transport, assembly, what have you, genes that actually make the Fimbriae. And so the way that these regulators, FIMB and E, work is that they're actually recombinases that work by flipping the promoter of the FIM operon so that FIME recombines from the on to off position so that it's facing the wrong way and you get no biofilm. Well, FIMB recombines from the off to on as well, and so it can flip the promoter in the right direction, and then you get biofilms and, all that good stuff. So you've seen these data before with the FIMB knockout, but one thing that's cool is that FIMB and E have opposite biological effects, and my predicted values show that they have roughly equal and opposite fitness effects over time at each time point. Now, this gets more complicated, though, if you look at the entire operon. Here's again FIMB and E on the left. The other knockouts are kind of all over the place. And one thing I haven't told you is that FIM-A actually encodes in its 3' UTR a small RNA, FIMR2, that regulates a whole host of metabolic processes as well as other stress response phenotypes like biofilm and motility and morphology. And so knocking out other genes in the operon likely not only disrupts biofilm, but also could have polar effects on disrupting this small, very important RNA. And so while it might feel kind of unsatisfying to end the talk on it's complicated note, the point is that transposon screens are very complicated. And so we're in this kind of exciting era of where computational methods and Bayesian inference computation has evolved to make a full Bayesian analysis tractable on such a large data set. And this kind of approach then is powerful enough to give this large data set kind of the careful interpretation that it deserves. And so to summarize, I am building this all into a pipeline R package that then can be used for just your average stats practitioner. It's kind of a drop-in replacement for what you would typically use to analyze these types of experiments. And in general, I've seen that it recapitulates a lot of biological effects that are already known. And I'd like to acknowledge the lab and my funding sources and my thesis committee and other mentors, and of course, the wife and dogs. Thank you very much. No, one thing I left out is that in general, my project has been looking at more feast-famine cycles, and so adding a fourth element to this of refeeding stress. And so the adaptations we see are slightly different than GASP, in that GASP is more of a hold on for your life, but this has to evolve towards a much more dynamic route. But yeah, hopefully I'd like to sort this out to get it more so we could use it on these cycles as well and use it for more than one experiment.
So, Carl, so kind of talking about what you were looking at the thin stuff, right? Especially when I'm thinking about we have these small RNAs and then have like four effects. We know that these transposable elements, right? We know PhD is knocked out, but there's multiple insertion sites across that entire library.
Mm-hmm.
Now that you've laid this annotation onto it, will you be able to have the nuance to kind of pick apart the fitness values of the different insertion sites, like ones that might interrupt both the gene and maybe small non-coding RNAs overlap, and ones that just like disrupt the gene?
Yeah, I mean, that's possible. It's a lot of it goes back to I didn't do the TN-seq mapping the barcodes. That was done by the original creators of the library. And I do have the position, so I guess I could re-annotate them all to, because as it is, only genes are annotated. But you could obviously annotate it however you want and then do those kinds of hypothesis tests just with this.